In this video, we are going to learn about paper electrophoresis. Two compartments or beakers containing buffer solution are interconnected by a supporting glass plate. The different buffer solution used in paper electrophoresis are also important. The level of buffer in the compartment is equalized. A suitable filter paper is subjected and is made into strips having the width of 3 to 5 centimeter. So electrode will be there, beaker is there, buffer solution is put into that, and the battery will be there in the middle. Sample is applied on the filter paper, supporting plate filter paper. Negative electrode and positive electrode are connected with the battery. So buffer solutions in electrophoresis: proteins, barbiturate buffer, borate buffer, phosphate buffer, and in case of amino acids, phthalate buffer, carbohydrate borate buffer. So barbiturate, borate, phosphate, phthalate, borate buffer. At the center of the strip, the origin line is marked with pencil in the form of a spot or marking. The paper is now soaked in buffer solution and pressed between the blotting paper. The sample is applied on the spot with the help of a capillary tube or micropipette without damaging the paper. Then the paper is placed on the supporting plate with each end dipped in the buffer solution of the opposite compartment. The electrode of the compartments are connected to a power supply. The current is applied and the electrophoresis is run for 45 minutes at a very low voltage. Then the separation is over, the paper is removed, dried and stained with locating reagents such as bromophenol blue, naphthalene black, ninhydrin, azocarmine, Schiff periodic acid reagent, diphenylamine, etc. Bromophenol blue, naphthalene black, and azocarmine are used for protein visualization. Locating agents. After illusion of fractions using a colorimeter, photoelectric scanning or a densitometer can make the quantification. The paper electrophoresis used for the separation of substance at low voltage is called low voltage electrophoresis. This method is valuable in the separation of high molecular weight substances like proteins, carbohydrates, and nucleic acids, but it has only limited value in the application of amino acids. When a high voltage current is used in paper electrophoresis, it is called high voltage electrophoresis, which is used for the separation of low molecular weight compounds and smaller ions like amino acids and inorganic substances. This technique requires a current of high voltage and longer duration. When the sample is allowed to flow continuously in the paper by some devices for separation, then it is called continuous flow electrophoresis. This method is useful in the separation of amino acids and proteins. Next one is the cellulose acetate electrophoresis. Here, cellulose acetate strips are used instead of paper to separate molecules with better resolution. While using cellulose acetate, the problem of adsorption is eliminated. Cellulose acetate is chemically pure, translucent, and not very hydrophilic. The buffer for this technique are the same as those used in the paper electrophoresis, but the solvents such as glacial acetic acid, cottonseed oil, liquid paraffin, etc., which make cellulose acetate transparent, are more preferred, like glacial acetic acid, cottonseed oil, liquid paraffin, etc. So cellulose acetate electrophoresis is used to separate glycoproteins, lipoproteins, and hemoglobin. In addition, immunoelectrophoresis uses cellulose acetate strips. Next is the gel electrophoresis. In gel electrophoresis, gels are used as the medium instead of paper. When the gel is placed as a column, it is called column zone electrophoresis. If the gel is coated on glass plates, then it is called open block electrophoresis. Column zone electrophoresis and open block electrophoresis. Column zone electrophoresis and open block electrophoresis. Disc electrophoresis is a column zone electrophoresis because gel is packed as a vertical cylindrical column in tubes. Next is the polyacrylamide gel electrophoresis. The apparatus consists of a buffer reservoir system and a power pack. The buffer reservoir system has an upper cathode compartment and a lower anode compartment containing buffer solution. Glass tubes having 1.4 cm length and 0.5 cm diameter connect the two compartments. The compartments are provided with platinum electrode. Buffer solution cathode compartment will be on the top and anode compartment will be on the lower side, and both of them are filled with buffer solution. Glass tubes are connected. Inside of that, packed gel will be there. One end of each tube is slightly closed with a rubber cork and is filled with 1.1 ml of small pore solution separation gel. Above the solution, 0.1 ml of double distilled water is added and allowed to polymerize for 30 minutes. After the removal of water layer, 1 to 2 ml of large pore solution spacer gel is overlaid, followed by 0.1 ml of distilled water for polymerization. After the removal of water layer, 0.2 ml of large pore solution spacer gel is overlaid. First we were adding small pore solution. Now we are adding large pore solution, and after that is overlaid, followed by 0.1 ml of distilled water for polymerization for 15 minutes. Then the water layer is removed and 0.05 ml of the sample is added, followed by 0.001 ml of marker dye. The remaining space of the tube is filled with the tris buffer. The preparation of the reagents and buffer is given. After removing the rubber corks, the tubes are attached to the compartments in such a way that one end of a tube is attached to the anode compartment and other end to the cathode compartment. Then the compartments are filled with tris buffer and current is applied. When the marker dye migrates to the required position in the tube, the electrophoretic run is stopped and the tubes are removed from the compartments. The gels are immediately removed from the tubes with the help of a syringe needle, and each gel is stored in 70% acetic acid contained in a test tube. The gels are stained with appropriate dyes and the sample is quantified spectrophotometrically or densitometrically. Next one is the two-dimensional gel electrophoresis. It's a powerful tool and designed by combining the resolving power of isoelectric focusing with STS page. As the molecular weight and isoelectric point of a macromolecule are not related with each other, this technique makes use of these two properties to separate the molecules with greater resolution power. By this method, a mixture of large number of proteins can be resolved into individual fractions. In this technique, a protein mixture is subjected to isoelectric focusing on gel in a capillary tube, in contrast to the conventional electrophoresis in which the pH between anode and cathode is constant. The isoelectric focusing, the pH gradient is maintained with gradual increase from anode to cathode. When the protein sample is introduced into the system at pH below its isoelectric point, it has a net positive charge and will migrate towards the cathode. Proteins possess net positive charge in an acid medium. Therefore, the protein molecule moves to medium with higher pH due to the presence of pH gradient. High pH of the medium influences the ionization charge of the molecule, so the number of positively charged particles decreases. Thus, at a particular pH or isoelectric point, the net charge of the protein becomes zero and will not move further. At this point, the gel is removed from the capillary tube. Keep on the sample wells of the slab gel and subjected to STS page. Now the proteins are separated according to their molecular weight. Gel electrophoresis is widely used to separate and isolate a large number of macromolecules. In molecular biology, this is a versatile technique used for determining sequences of DNA molecules, the nature of nucleic acids with the restriction maximum of DNA, etc.